Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing DBT BET JRF Entrance Exam Question Paper Part 2 for the year 2022. Part 2 I have divided it into two parts, that is Part 1 and Part 2. Part 1 I have already uploaded. You can see the video in my channel. So if you are new to my channel, please do like, share and subscribe. Then, which of the following statements are correct? Lipid bilayer lacking protein is highly impermeable to all charged molecules. That's correct. Channels have specific binding pockets for the solute molecules they allow to pass. So specific binding pockets, it is coming under transporters and not channels. So that is not correct. The next one. Transporters allow solutes to cross a membrane at much slower rate than do channels. That is correct. The plasma membrane of many animal cells contain open K-positive channels, yet the K-positive concentration in the cytosol is much higher than that outside the cell. That is also correct. The next one. The membrane potential of an axon temporarily becomes more negative when the action potential excites it. It becomes more positive, not negative. So E is not correct. So A, C and D are correct and your answer is option 1. The next one. Which one of the following enzymes help the DNA to overcome the tension generated by the unwinding during replication? So naturally it is the topoisomerase. Answer is option 3. The next one. Which one of the following types of mutations can lead to a major change in the encoded protein? So insertion of a single nucleotide near the end of the coding sequence. That will not lead to major change in the encoded protein. Removal of a single nucleotide from the beginning of the coding sequence. Yes, it can lead to frame shift mutation. So answer is option B is correct. Then deletion of 3 consecutive nucleotides of a codon in the middle of the coding sequence. That also will not result in major change in the encoded protein. Only one amino acid will not be there. Then next one. Deletion of 4 consecutive nucleotides in the middle of the coding sequence. That of course will lead to a major change in the encoded protein. Again, some frame shift mutation can take place. So the answer here is option 2, that is B and D. Then next one. Which one of the following organism is not an obligate intracellular parasite and therefore can grow on bacteriological media? So it is mycoplasma. Answer is option 1. Then next one. Differential expression of the genetic material depending on its parentage of inheritance is known as? It is known as genomic imprinting. Then which of the following statement is most relevant to leishmaniasis? Most relevant they have asked. Large domestic animals such as cattle are the principal reservoirs of Leishmania donovani. It is not cattle, it is dogs are the principal reservoirs of Leishmania donovani. Both visceral leishmaniasis and cutaneous leishmaniasis are transmitted by the bite of sandflies. That is correct. Artemisinin is effective in the treatment of leishmaniasis. Amphotericin B is effective in the treatment of leishmaniasis. So that is also not correct. Leishmaniasis is transmitted by the bite of female Anopheles mosquito. That is also not correct. So the correct answer here is option B. Then next one. Which form of plasmodia is transmitted from mosquito to humans? It is sporozoite. First one is the answer. AIDS is caused by human retrovirus that kills CD4 positive T lymphocytes. Answer is option 3. Then latency is characteristic of which of the following viruses? It is herpes virus. Answer is option 1. Then next one. Which one of the following is the drug of choice for sexually transmitted diseases, urethritis and cervicitis caused by chlamydia trachomatis? So it is azithromycin. Answer is option 4. Then next one. Which one of the following is the most accurate statement regarding giardiasis? The drug of choice for giardiasis is chloroquine. It is nicotinamidazole drugs, that is metronidazole, tinidazole, ornidazole. These are the drugs, not chloroquine. Then Giardia lamblia produces an enterotoxin that increases the CAMP within the enterocyte, resulting in diarrhea. That is also not correct. Giardia lamblia infection is acquired by ingestion of food or water contaminated with human feces. That is, there is no animal reservoir. There is animal reservoir for this organism. So 3 is also not correct. Fourth one, if you see, infection by Giardia lamblia occurs primarily in the small intestine, frequently resulting in the malabsorption of protein and fat. That is correct. Option 4 is the answer. The next one. T cell clones in an individual recognize peptides only when bound and displayed by that individual's MHC molecule on the surface of antigen presenting cell. This process is known as MHC restriction. Answer is option 2. Then next one. A bacterial signaling system that regulates population density dependent gene expression using secreted chemical signals is known as quorum sensing. Answer is option 2. A probiotic is a live organism that is claimed to confer a health benefit by altering the indigenous microflora of the intestinal tract. Which one of the following bacteria has been widely used in probiotics? So it is Lactobacillus lactis. Answer is option 3. Match items in list 1 with items in list 2. Choose the correct answer from the options given below. Down syndrome is fourth one, XX21. Edwards syndrome is trisomy of 18. Klinefelter syndrome is XXY. Turner syndrome is female with only one X. So the answer here is option 3, A4B1C2D3. The next one. The animal embryonic stem cells that can differentiate into an embryonic cell type are known as pluripotent stem cells. Then RTS vaccine for malaria consists of central repeat R region derived from Plasmodium falciparum, circumsporozoite protein that is genetically fused to the T cell epitope, and surface antigen S regions derived from hepatitis B virus. Answer is option 2. Then next one. Short chain variable fragments SCFV of antibody are composed of. So SCFVs are recombinant antibodies and they are approximately 25 kDa single polypeptides containing VL and VH of an antibody. These two chains are connected by flexible linker peptide that is usually 15 to 20 amino acids long and made up of glycine and serine. So the answer here is option 3, that is VH and VL domains. Then next one. Proteins used for the purification of IgG is isolated from. So they are isolated from Staphylococcus aureus. The next one. In a neuronal culture experiment, the effect of the chemical under study was prevented by pre-treating the cells with parazosine, an alpha-1 adrenoreceptor antagonist, but not by propranolol, a beta-adrenoceptor antagonist. The chemical under study is likely to be A. So it is a noradrenaline or methoxamine. Answer is option 4. Then which one of the following is not applicable for a chemical synapse? So it cannot modulate signal intensity. Answer is option 3. Then next one. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. So assertion A. Heating a pre-optic anterior hypothalamic area of the brain caused profuse sweating and dilation of the blood vessels of the skin. The above in A is to absorb more heat into the body and throw out waste from the body. So out of these, A is correct, but R is incorrect. So the answer here is option 3. Then sleep loss may induce. So it may induce increased memory loss, compromised immune system and decreased cognitive ability. So answer here is option 4. Then next one. Which one of the following is not a function of the glial cells in the brain? So transporting neurotransmitter vesicles from soma to terminal. That itself is the answer. So option 1 is the answer. That is not the function of glial cells in the brain. Then next one. Scotomorphogenesis is characterized by. So the development of seedlings in the dark. It is called as scotomorphogenesis. So small and closed cotyledons and elongated hypocotyle. So that is characteristic of scotomorphogenesis. Then node gene products function as receptors of certain flavonoids. So node factor synthesis is directed by rhizobial node genes. And node factors secreted by rhizobia initiate the process of nodule formation in the legumes. So node gene products, they function as receptors of certain flavonoids. Then next, the ligand binding domain of a ligand gated channel has a serine residue, phosphorylation of which is essential for ligand binding. By side-directed mutagenesis, it was replaced by aspartic acid. The likely consequence will be. So aspartic acid, it is chemically similar to phosphoserine. So what they have told here is the ligand binding domain of a ligand gated channel has a serine residue. Phosphorylation of this serine residue is essential for ligand binding. Now by side-directed mutagenesis, the serine phosphorylated serine was replaced by aspartic acid, or serine residue was replaced by aspartic acid. So the likely consequence will be. Now aspartic acid, what is it? It is a phosphomimetic of phosphoserine. So once serine is replaced by aspartic acid, it can make the protein always in the phosphorylated form. So once it is always in the phosphorylated form, the ligand gated channel, it becomes constitutively active. So the answer here is option 2. Then next one. Cryptochromes control photomorphogenesis in response to. So cryptochromes are blue light receptors. So blue and UVA light. Answer is option 4. Then which one of the following is an important constituent of RNA-induced silencing complex in plants? So it is argonate. Answer is option 1. Then next one. miRNAs are major regulators of gene expression. In an experiment, it was observed that gene X is a direct target of miRNA-A. Upon overexpression of miRNA-A in Arabidopsis thaliana. So in the experiment, it was observed that gene X is the direct target of miRNA. Gene X is the direct target of miRNA A, and you have overexpressed miRNA A in Arabidopsis thaliana. So both, what will happen is, upon overexpression, both transcript and protein abundance of X may get reduced. Then next one, Vir A and Vir G form a two-component signaling system during agrobacterium infection in plants. In an experiment, Vir G was mutated, resulting in non-functional Vir G protein. Which of the following consequence is most likely to occur? Now, Vir G, it is a phenolic signal response regulator to activate other Vir genes. So now Vir G is mutated means phenolic signal will be perceived but not transduced. Phenolic signal will be perceived because Vir A is functional. So phenolic compounds, they interact with Vir A protein, which is a phenolic signal sensor. So phenolic signals will be perceived but they will not be transduced. So the answer here is option 2. Then 1 centimorgan is defined as the genetic distance between two loci with a statistically correct recombination frequency of 1%. Answer is option 3. Which of the following genes can be used to generate male sterile crop plants? So it is the Barnase. Option 1. Then which one among the following is the correct function of leg hemoglobin? So it sequesters oxygen to facilitate nitrogen fixation. Answer is option 1. Then which one of the following is most commonly used as a selection marker for developing transgenic plants? So it is hygromycin phosphotransferase. The others are all scorable markers, reporter genes. Selection marker is hygromycin phosphotransferase. Then in plants, the overexpression of lectin genes confers resistance to insect. Answer is option 2. Then next one, a spectrum that depicts magnitude of response of a biological system to light as a function of wavelength is called as, so it is called as action spectrum. Action spectrum indicates the overall rate of photosynthesis at each wavelength of light. So here magnitude of response of a biological system example will be photosynthetic rate. So the action spectrum, it indicates the overall rate of photosynthesis at each wavelength of light. Whereas absorption spectrum, it indicates the wavelength of light absorbed by each pigment, that is chlorophyll and carotenoid. Then next one. In a metabolic engineering experiment, the flux from primary carbon metabolism was diverted towards methyl erythritol phosphate pathway, MEP pathway. Which one of the following class of secondary carbon compounds do you think will accumulate more? So MEP pathway, it is responsible for the biosynthesis of isoprenoids, and terpenes is an example of isoprenoid. So answer will be option 4. Then next one, a haploid sperm from one species and a haploid egg from another species may form a diploid interspecies hybrid. Meiosis in these plants generally fails but can also lead to rare duplicated gametes called allopolyploids. So allopolyploids, they occur when an polyploid offspring is derived from two distinct parental species. And autopolyploid, it occurs when polyploid offspring is derived from a single parental species, usually via self-fertilization. So the answer here is option 1. Then, which one of the following is not a typical feature for feature of the hypersensitive response observed in plants during the attack of invading microbes. So rapid spike in photosynthetic rate. Answer is option 3. Then next one. Given below are two statements. Large subunit of Rubisco is encoded by the chloroplast genome, while small subunit is encoded by the nuclear genome. That is correct. Small subunit of Rubisco has a transit peptide S, which is cleaved off at the time of its translocation into plastids. That is also correct. So both statements 1 and 2 are true. Answer is option 1. Then next one. The most likely subcellular localization of NAC transcription factor is. So the NAC family of protein is one of the largest plant species transcription factor family, and its members play varied roles in plant growth, development and stress response. So the most likely subcellular localization of NAC transcription factors is in the nucleus. So answer is option 1. Then next one. In plant transformation vector, the selectable marker gene is ideally located next to the left border of the tDNA region because left border of the tDNA enters last into the plant cell. That is correct. Right border of the tDNA, not right border, left border. Then it ensures complete transfer of tDNA into plant cells. So A is also correct, C is also correct. So answer is A and C only. Then next one. Given below are two statements. One is labeled as assertion and the other is labeled as reason. Choose the correct answer from the options given. Phosphomannose isomerase from E. coli was used as selection marker to develop golden rice 2. When mannose is added in the selection media, endogenous hexokinase converts mannose into mannose 6-phosphate, which block glycolysis, ATP production, and represses transcription of photosynthetic gene, resulting in slower growth of non-transformed plants. Phosphomannose isomerase, on the other hand, convert mannose 6-phosphate to fructose 6-phosphate. So both A and R are true, and R is the correct explanation of A. Answer is option 1. Then I have not answered questions from bioprocess technology. So coming to question number 109. For determining Vmax and Km of an enzyme catalyzed reaction, we often use a double reciprocal plot of 1 by V versus 1 by S. This is not the best method because why Lineweaver-Burke plot is not the best method always, they have asked. So that is because errors increase for smaller values of V and S. Then next one. For monoclonal antibody production, mammalian expression systems are preferred over bacterial expression system because mammalian system provide necessary post-translational modifications. Answer is option 3. Then next one. Yeast cells obtained from batch fermentation can be reutilized for the production of beer. Answer 1. Then next one. Which one of the following is not a food rich in vitamin A? So potato is not a food rich in vitamin A. Answer is option 1. Then next one. The principal microorganism for yogurt is, it is Streptococcus thermophilus. Answer is option 1. Then the red color of beetroot is due to the presence of, so beetroot it is red in color because of the presence of betalains. Answer is option 1. Then next one. Commercial production of citric acid is done by fermentation using, so it is Aspergillus niger. Answer is option 1. Given below are two statements. One is labeled as assertion A and the other is reason. The light of the above experiment, choose the most correct option. The dot matrix method of sequence alignment is very basic method that should be used first before other more complex methods. That is correct. The diagonal dot matrix display can readily reveal presence of insertion, deletions, and direct and inverted repeats that are more difficult to find by other more automated method. That is also correct. So both and R is the reason for A also. So both A and R are correct, and R is the correct explanation of A. Then next one. Given below are two statements. Statement 1 and statement 2. Choose the most appropriate answer from the options given below. An alignment is generated by starting at the ends of the two sequences and attempting to match all possible pair of characters between the sequences, and by following a scoring scheme for matches, mismatches, and gaps. That's correct. Statement 2. For proteins, an amino acid substitution matrix such as day of person accepted mutation matrix 250, that is PAM 250, or blossom substitution matrix 62, blossom 62, is used to score matches and mismatches. That is also correct. So both statement 1 and 2 are correct. Then next one. Given below are two statements. Statement 1. RNA secondary structure is composed primarily of double-stranded RNA regions formed by folding of the single-stranded molecule back on itself. That is correct. Like, say statement 2, like protein secondary structure, RNA secondary structure can be conveniently viewed as an intermediate step in the formation of three-dimensional structure. Statement 2. So both statement 1 and 2 are correct. Answer is option 1. Then next one. A protein sequence has 20 consecutive hydrophobic amino acids followed by few hydrophilic residues, and this pattern repeats itself 5 to 7 times. Which kind of protein is described here? Your GPCR is an example of it. So it is a transmembrane protein. Answer is option 2. Then next one. Which one of the following does not represent known interactions of RNA secondary structural elements? So it is helix turn helix. It is present in transcription factors. So the answer here is option 4. Then next one. Which one of the following can be considered as data matrix? So marks and ages of students in a classroom. Answer is option 2. Then next one. Which one of the following can be considered as an atomic vector? So that is your name can be considered as an atomic vector. Then next one. Which one of the following lines of Unix commands can tell you the number of lines in a file? So it is WC-L. Answer is option 4. And then next one. Which one of the following is not a data type? So thread is not a data type. Answer is option 4. Then which one of the following is comparatively the best e-value to a pairwise sequence match? So smaller e-value, then more likely the match is significant. So IE raised to minus 4 is the answer. Answer is option 1. Then next one. Your friend runs a blast P on his unknown sequence and find a match with e-value of 3 with the target. What does this mean? So e-value, it is the number of hits one can expect to see just by random chance. So there are 3 hits of similar score that can be found by random chance. So answer is option 4. Then next one. Where is the greatest likelihood of the occurrence of a proline residue in a protein structure? So it is in the turns and the loop. Then next one. Where is the greatest likelihood of the occurrence of cysteine residues in a protein structure? In the active site pocket. Answer is option 1. Next one. Which family of protein has the greatest likelihood of containing a leucine zipper motif? So leucine zipper motif, they are found in DNA binding motifs. So it is your transcription factors. Answer is option 3. Then next one. Protein usually have hydrophobic interiors or cores and hydrophilic exteriors. If a protein comprises hydrophobic patches on its outer surface, what can it mean? So it could be a multimeric protein complex. So if a protein has hydrophobic patches on its outer surface, it means it is attached to another protein. Okay, so it could be a part of a multimeric protein complex. Answer is option 3. Then during analysis of next-gen sequencing data, what is the advantage of BAM files over SAM files? So BAM file is binary alignment map and SAM is sequence alignment map. And BAM files are in binary file format which is not readable by humans. So BAM files are human readable while SAM files are not. It is SAM files that are human readable and not BAM files. Then BAM files are larger than SAM files. BAM files are not larger but they are smaller than SAM files. Then BAM files are smaller than SAM files, hence easier to transfer. That is correct. So answer will be option 3. BAM files can hold more information than SAM files. That is also not correct. So what is the advantage of BAM files over SAM files is that BAM files are smaller than SAM files and hence easier to transfer. The next one. The measure of similarity between two structurally aligned macromolecules is. So it is root mean square deviation. The next one. Which command is suitable to list out all the directories in the working directory in Linux Unix environment? So it is option 3. The next one. Which region of the gene, which of the following is true for PAM 250 scoring matrix? It is used for structural database search. It is used to score sequence alignments. It is based on an evolutionary model that predicts the type of amino acid changes over long periods of time. That's correct. Option 2 is correct. Next question. Which of the following cell separation techniques allow cells from different stages of cell cycle to be separated simultaneously with minimal perturbation to their physiology? So it is counter flow centrifugal elutration. Answer will be option 1. Then next one. Choose the correct statement in respect to metazoan development. So delta is a cell surface protein while notch is an intracellular protein. Notch is a transmembrane protein. Notch is a cell surface protein. That's also not correct. It is a transmembrane protein. Both delta and notch are present on the same cell. That is correct. Option 3 is your answer. And next one. The technique used in animal biotechnology for rapid multiplication and production of animals with their desired genotype is. So it is in vitro fertilization and embryo transfer. Option 3. Then next one. In animal cloning protocol, while performing enucleation, which chemical is used for relaxing the cellular cytoskeleton? It is cytochalacin B. Arrange these soluble cations in ascending order of the abundance of soluble cations found in saline water bodies. So it is Na greater than Mg, greater than calcium, greater than potassium. Answer is option 1. Then next one. Which one of the following process is not used to convert the hydrocarbons present in organic compounds to certain metabolic intermediates? So it is extraction. Extraction is the phase for separation of minerals from their ores. So answer here is option 2. Then next one. In a water sample collected from an industrial cluster, the ratio of BOD is to COD is found to be less than 0.3. Which one of the following may be presumed from this initial data? There are numerous carbon-based compounds in the sample which are not utilized by microbial population. That is correct. Answer is option 1. Then next one. Given below are two statements. Fungi are important degraders of polymers and are used in the composting and biodegradation of toxic organic substances. Statement 2. Fungi are able to selectively excrete toxins present in these polymers. Statement 1 is true. Fungi are important degraders of polymers and are used in composting and biodegradation of toxic organic substances. That is true. But statement 2 is false. Fungi are able to selectively excrete toxins present in these polymers. Statement 2 is false. So statement 1 is true but statement 2 is false. Answer is option 3. Which of the following methods can be used to treat crude sewage and nitrify secondary effluents? So it is reed beds. Answer is option 2. Then identify the correct statement about fucoidans. So fucoidans are sulfated polysaccharides found in brown algae that contain L-fucose among their constituent sugars. Answer is option 4. Then next one. Due to the difficulty of isolating and culturing, many marine microorganisms are regarded as viable but non-culturable. Which of the following is suitable method to hunt for useful genes in these organisms? So it is metagenomic analysis. Answer is option 4. Then next one. Ground shark cartilage ointment is popular for its role in fast wound recovery. The active ingredient primarily responsible for this function is N-acetyl glucosamine. Answer is option 1. Then next one. Which one of the following is not extracted from seaweeds? So calcium is not extracted from seaweeds. Then next one. Choose the correct option to fill up the blanks. Dash nourish the host coral as well as help it deposit its skeleton. They perform dash and pass on some of the dash that make the coral. So it is zooxanthellae nourish the host coral as well as help it deposit its skeleton. They perform photosynthesis and pass on some of the organic matter to make the coral. So answer is option 4. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.